Welcome to the joint meeting of the American Society for Microbiology's 55th Inter-Science Conference on Antimicrobial Agents and Chemotherapy and the International Society for Chemotherapy's International Congress of Chemotherapy here in beautiful San Diego. For those of you joining us from the web or for those of you joining us for the first time, welcome. This is a meeting whose focus is on the cutting edge of new antibiotics, antibiotic resistance, and the concept of antibiotic stewardship. This joint ICAAC meeting presents an unmatched opportunity for discussion and cross-collaboration amongst our globe's leading scientists and healthcare providers. I'm Michael Schmidt from This Week in Microbiology, and the organizers have asked some of the people presenting one of the over 1,600 abstracts here at this meeting to come and join us here at ASM Live. All you need to do, if you have a question, is present yourself to the microphone and we'll be happy to take your questions. Those of you in the Twitterverse, please use our hashtag ASMLive and we'll be happy to entertain your questions or from the ASM chat room. So I'd like you to welcome our two guests here this morning, Dr. Nishal Govindji-Bhatt and Dr. Curtis Dobson, who are joining us, and we're going to be talking about some really cool things. Dr. Bhatt presented their collective poster yesterday in the open session, and she had a large crowd that was interested in learning, and so I get this right, about a Microbiosensor, a device for monitoring bacterial contamination in contact lens cases. So Dr. Bhatt, why don't you share with us the premise of what question did your study ask?
So the main focus of our research, the aim of what we were trying to do here, was to reduce the overuse of antibiotics. And we're doing that by targeting or trying to reduce infection, get it early on, so that people don't need to have antibiotics.
So I guess it's fair to say we've been working for a number of years in the area of contact lens infection, which goes back about 20 years in my case. So historically we've been interested in trying to find better biocides to prevent contact lens infection, but a few years ago we started looking at whether there's an alternative, which is to detect infection early. And essentially, if there's a way of flagging that to patients, then potentially they can intervene at a time when normally they wouldn't be able to. And by doing so, they can throw away their lens cases or their solutions and potentially prevent an eye infection, which in some cases can be quite serious. And ultimately, that has impact on antimicrobial stewardship because we prevent infections that don't need to happen, essentially. So that's the premise. And the idea was to develop a sensor that would fit inside a contact lens case and would give a very simple, unmistakable readout to the consumer. They wouldn't need training. It would be an all-or-nothing output. It wouldn't go off again if it triggered, so that consumers were alerted to a problem. And I think this feeds into the whole area of contact lens compliance with the normal protocols for using contact lenses, and this is a major issue in the industry, and I think our technology potentially helps address that, or helps to improve that. That's an overview.
Yeah, one of the things I think when you're a contact lens wearer, you often forget, even though your healthcare provider tells you is the most important thing you can do is wash your hands before interacting with your lenses, because our hands are wonderful objects for transferring microbes, and of course we flick the lens out of our eye and we drop it into this case in which there is this solution, and we have no way of understanding whether or not the solution is already hot. Those of us who are microbiologists, of course, know that when in the laboratory we have this beautiful readout of bacteriological media. We know when it's bad because it becomes turbid. Unfortunately, the average consumer can't appreciate when there's substantial numbers of microbes in their case. And the other flip side is you don't need very many to cause an infection of the eye, and so it doesn't achieve turbidity in the case.
No.
So the consumer doesn't have that opportunity. So how did you go about thinking about a biosensor that a consumer could immediately appreciate?
Sure. Well, I think you're right. Certainly in terms of the levels, that was a key thing for us to think about. And actually, contact lens solutions can go turbid at about 10 to the 6 CFU per mil. So if you have about 1 million infecting organisms in a mil of liquid, that is actually visible to the patient. But actually, you can get a nasty infection from much lower levels of contamination. In fact, typically about 10 to the 3 is the level where patients are asymptomatic. So if you have about 1,000 infectious organisms per mil, you don't tend to notice symptoms for most patients. So we targeted that sort of middle zone. And if you want to talk about how you did that, but essentially we've developed the device so that we can target about 10 to the 4, so about 10,000 infecting organisms per mil is where our device triggers universally. There's a little bit of variation from species to species, but essentially around there. But I think that was the idea. I mean, now putting it in the case was you can come back to it in a second. Just another broad point. Putting it in the case was a decision we took again to keep things simple. We envisaged at one point trying to put it in the lens itself, but there could be issues if the device changes color. It could affect your vision, for example, and also people might have to look in the mirror to see that it had triggered. And actually, the highest risk is probably just after an overnight disinfection. If the disinfection hasn't worked, that's the time you want to know about it before you put it back in your eye. So I think the case seemed to be the sensible place to choose.
And Curtis is quite right, and as you were saying as well, the case is the best place because it tells you whether or not the contamination is from the case, it tells you whether or not it's from the multi-purpose solution, and it can tell you whether or not it's from the contact lens itself. It targets all 3 areas.
So they will converge on that point.
That's right, so it will tell the end user one of those 3, if not all 3 or 2, means that you don't use that contact lens in your eye.
Is it a very generic indicator, so it can pick up any type of microbe that may be in that solution? So what organisms did you specifically target this sensor to appreciate?
So with bacteria, about 80% of the causative organisms are Pseudomonas, Staphylococcus and Streptococcus. We have a standard that's commonly used in the contact lens area, and they say that the 5 common organisms are Pseudomonas, Staphylococcus, Serratia, Candida albicans, and Fusarium. So there's 2 fungi in there, 3 bacteria, and they are the ones that we've been looking at. There are, of course, other microorganisms, protozoa, like Acanthamoeba, that also cause infection, but we concentrated on the top 5 there.
Yeah, that's the thing I always worry about is the Acanthamoeba, because I see when I was a teenager, some of my friends who were wearing hard contact lenses would routinely take them out and run them around in their mouth and then actually put them back into their eye, and the Acanthamoeba would come along with them.
Sure, yeah.
And so that would always just freak me out as they re-wetted their lenses by using good old saliva.
And typically Acanthamoeba is associated with swimming in contact lenses, so there are other ways, and tap water is another one. So we haven't formally tested Acanthamoeba, but theoretically Acanthamoeba should be just as likely to trigger the sensor as other organisms. So, and I mean, another thing to bear in mind is Acanthamoeba is often associated with other organisms colonizing the lens first, because it often uses bacteria as a food source. So again, that just detecting bacteria is a good way of demonstrating whether or not Acanthamoeba will be supported in that system. So the one group of organisms that we don't target is viruses, because certainly for contact lens microbial keratitis, viruses are not considered to be a major cause. I think for broader microbial keratitis, herpes simplex virus can be a cause, but in our case, we didn't think that was important for contact lenses. So that's one differentiating feature. So essentially anything that's not a virus, our sensor will pick up.
So you had an interdisciplinary team that worked on this. In addition to you two who were on the poster, we had Andrew McMain, Phil Morgan, and Nicholas Goddard, who were all co-authors. So why don't you just give them their proper credit and the contribution that they made, because I think we need to appreciate that this is just not a purely microbial study that this was an interdisciplinary team. You had the leaders and the ophthalmic industry looking and assisting you to come up with a really solid solution for the consuming public.
Sure. So, I mean, I can talk a little bit about the history of that. Essentially, the group has worked for a very long time with the EuroLens group, which is a major clinical trials group in Manchester University. And Phil Morgan and Carol Maldonado-Godina are the kind of two leads in that group. So they're practicing clinical optometrists. And I had historically had quite a number of links with them through my work on biocides. So we were developing antimicrobial peptide biocides for use in contact lens solutions with them. And that work continues through another vehicle at the moment. So that was the starting point. And then we, literally about 5 years ago, when we decided to try and attempt this approach, we assembled some other colleagues from around the university that had expertise that could help us. So Andrew McBain is a microbiologist who I think you've had some connections with in the past. He runs a biofilm group in the university, so they're a very well-known biofilm group. Nick Goddard is a mechanical engineer, essentially works on microfluidics and manufacture of small devices. So one of the key things for us was to not develop something which wouldn't be commercializable, that wouldn't be useful. We wanted to have something that wasn't just an interesting research toy, but actually something that you could potentially manufacture on a massive scale at a reasonable price. So that was important to get Nick's involvement in that. So we designed something that we could actually commercialize. So do you want to have any comments?
Well, I'm just going to ask a question. We have a picture of your image that will go out on the web. So Chris, could you please push that to the net? And so we have your image up there of what this looks like. Okay. So why don't you describe how the sensor works? And you're going to have to do it like you're on radio to the audience here, because they unfortunately can't see the image. But those who are watching on the web can of course see your magic. So we'll see how well you can describe it to our audience, and they may ask you a question. Oh, I should also point out to the members in the audience, if you have a question, just please come up to the microphone. We'll be happy to take your questions. And Dr. Govindji-Bhatt, why don't you just take us through a little bit about how this wonderful sensor actually detects for the presence of microbes.
So I think the image that you're looking at the moment is 5 contact lens cases, and at the bottom of the contact lens case there's a panel. The top panel has got a straw yellow colour, and the bottom panel there has got a dark blue or black colour. So it's turned around a little bit, but if you just imagine that for this photo that you're seeing, one side is showing no contamination, so no infection there, and the other side that's black is showing that there is the presence of microbes. So our technology is ordinarily a yellow colour. The chemistry in there is a yellow colour. When bacteria or microbes are present, they will metabolize the chemistry there and turn it into a dark blue colour. So the end user, when they wake up in the morning, they go to put their contact lens in their eye. If they see this dark blue colour, they'll know not to do that. So it really is a very, very clear signal. Even for somebody that does wear contact lenses, obviously their vision won't be great, so they can still see this obvious colour change.
And it's not subtle. It looks like a button has been placed in the bottom of the swell. It's really, it's black or white. And
that's right, yeah.
It's really very, very simple. So the average consumer just needs to look at it and say, is there a button in the bottom of the well? And if there is, throw it out.
That's right. I mean, most people will go there, they'll start off with contact lenses. They'll be taught this is how you're supposed to look after your contact lens. You're supposed to rub and rinse, they call it. So you take your contact lens out, you rub it in multi-purpose solution before you go to sleep. You take it out, rub it in multi-purpose solution, put some multi-purpose solution, which is a disinfectant, in your contact lens case, put your contact lens in there. When you wake up in the morning, wash your hands and do the same again. You take the contact lens out, you rub it with multi-purpose solution, you throw away the multi-purpose solution that's in the contact lens case, and off you go. along with your day. And most people along that process, that routine, will do a number of things wrong. They won't wash their hands. They won't probably use multi-purpose solution. They might, as you said, use their saliva or tap water or saline, because they don't realise what the function of the multi-purpose solution is. Other people won't rub and rinse properly, or they'll rub and rinse
with
in tap
dirty
water.
hands.
Exactly. So this is all non-compliance, and our device is there as a backup. Most people will go for many years and nothing will happen to them, and they'll think, well, I'm fine, I've been doing it this way for many years. But it only takes that one time when you do have a high level of microbes present, either in your multi-purpose solution, your contact lens case, or on your contact lens, when you're in serious trouble and you could lose your vision. Our device is there to just give them that reassurance that yes, you're doing everything fine and you're okay, and give them a backup to, when it does change colour, not to put it in their eye.
So after they use your case and they dump the contents out, do they just leave it air dry and it's reusable over and over again?
Well, people will have their contact lenses for a month, and after that they're supposed to throw everything away. They're supposed to
The case as well?
The case as well. So the multi-purpose solution will come with a case, and you're supposed to have a new contact lens, new multi-purpose solution, and a new case, change it every month. Some people don't do that. They think, well, I'm not going to spend my money, for example. I'm going to keep hold of what I've got and use it for another month, and see how it goes. They're really not supposed to do that.
Some people do that for years, don't they?
Yeah.
Your husband, you were saying.
Yeah, well, yeah.
Don't pick on your poor husband. He's not here to defend himself.
But a lot of people, like I was saying, they feel they've never had a problem. So why not just do all these other things that they're not trained to do at the beginning?
Well, have you begun to look at the cost associated with this? Does it add significant money to the price, or is it the value and the cost are so inexpensive, or the value is so high and the cost is so low that it's a no-brainer to
Well,
do?
let's put it this way. In the US alone, about 1 million people will go to their practitioner with problems associated with their contact lens wear, and it costs the US about $175 million a year, infection itself associated with contact lenses. So if we can reduce that, then I think we're onto something.
And would either of you like to talk about the keratitis? I mean, the listening audience out on the internet may not know the severity of this sort of infection and what they might anticipate if they fail to wash their hands or to care for their lenses properly.
Sure. Well, a number of points there. I mean, one more reassuring point is at least it's very rare. It's pretty rare. But at the same time, so many people use contact lenses, that means a number of people do get affected every year. So, I mean, I think the sort of statistics that we typically quote, I think 6,000 people per annum lose some form of visual acuity every year from contact lens-associated infections. So microbial keratitis is the most serious form of adverse response to use of contact lenses. So, and for a lot of optometrists, they're effectively a kind of high street shop. You know, it's a consumer product. They don't normally have issues like this at all, but very occasionally they'll have a patient with microbial keratitis who will lose their vision. You know, so for them it's a really major problem. It's very rare, but when it happens, it's dreadful. So, you know, so certainly, and certain types of infection, particularly pseudomonal infection or acanthamoebal infection, are very aggressive and often very difficult to treat. So, by losing visual acuity, it can range from sort of scarring of the cornea through to the actual eye being removed. So, you know, very rare, so we don't want to alarm people, but at the same time, occasionally the consequences can be very severe. So our aim is to, you know, prevent that, is to, you know, reduce that significantly, I think. So
people generally, when they experience discomfort with wearing contact lenses, it's like having sand in your eye. Our eyes are really, really sensitive. So it's like having sand in your eye. It's like a gritty feeling, and it can't be very pleasant. That is a form that it's telling them that there's something not quite right with their eye. There is some kind of inflammatory response there, and that can be caused by bacteria that's present on the contact lens. It can be caused by other things as well, small scratches on the cornea, dryness.
So in addition to these kind of serious, dangerous infections, as Nishal's alluding, that there are often a number of inflammatory responses that can come not from an acute infection of the cornea caused by contact lenses, but by the presence of microbes on the lens or in the lens case. And those are not quite as dangerous, but they're still very uncomfortable. They can lead to things like contact lens peripheral ulcer and contact lens red eye. And again, the patient might need to discontinue use of the lens, go and visit the ophthalmologist. So again, those aren't kind of medical emergencies, but again, our technology has the potential to reduce those, because again, if you can lower the number of infectious organisms by effectively having zero tolerance on colonization in the cases, then potentially you can eliminate or significantly impact some of those inflammatory conditions. So I guess there are two sort of key aims. There's the very dangerous but rare microbial keratitis, and the more common but uncomfortable and not quite so dangerous inflammatory events that we'd like to target.
You're watching ASM Live at ICAAC, and if you have any questions, I encourage you to post them to at ASM Live, or if you're in the audience here in San Diego, please feel free to come up and ask a question. We're visiting with Dr. Nishal Govindji-Bhatt and Dr. Curtis Dobson about yesterday's poster here at ICAAC. So one of the questions I have is, this may be a little bit premature, but are we getting ready to manufacture this biosensor? What's the commercial development look like? It sounds, or do we go to a clinical trial next? So what are your plans? It's always, you have to answer the question, what's next?
Sure,
and so
sure.
What are your plans?
I think the next stage for us is clinical validation. So I think we're well placed to do that. Now that can be done ex vivo initially, so we don't necessarily have to use the device in anger with patients who are routinely using lenses. We can take samples from lenses that have been worn and so on. So I think that's the next step. It is an interesting proposition commercially. Contact lens cases are normally given away with multipurpose solutions. So in a sense, we either get into a sort of an arrangement with an existing manufacturer and provide these cases as part of that package, or alternatively provide these as a product which could be prescribed or used by optometrists in the first instance. I think maybe that will be the way to go in the first stage, because there are populations who are particularly well placed to benefit from the technology. There are demographics that are prone to eye infections. It actually typically tends to be sort of young male adults seem to be one of the higher risk groups, for example. So if you're an optometrist, you might want to recommend, if you had a young male adult who has a history of inflammation or soreness with using contact lenses, you might want to recommend they would use the case first. So I think for us that might be the next place to go once we have approval for use, would be to look at a product that's positioned in that way. Ultimately, this could be used by any daily wear user, of course, though in the longer term, so that any daily wear user could use this as part of their normal care routine. And ultimately, that might be the best way of using this, so that essentially we have zero tolerance across the board on colonizing organisms in cases, which would be great if we could do that.
One of the attractive things is it's when you begin to use contact lenses for the first time, you're just so excited to get this new product to shed your glasses.
Sure.
And patient education is very essential because you're literally placing a device. And those of us in the medical microbiology arena know that medical devices, the only thing that bacteria like better than people are medical devices.
Indeed.
And so they quite frequently attach, and so consequently any way that we can have an alert that things have may gone awry would be, I think, just tremendous.
Sure. I think that was our aim, and we certainly take the point that across medical devices this is a kind of ubiquitous problem. In fact, my research group is the medical device biology group, and we focus on medical device infection, not just in ophthalmics, although ophthalmics is one of our key areas. So I think we see this as a technology that has applications for other devices too, I think, and things like wound care products, one of the areas that we've been looking at more recently. So yes, I think you're right. I think if we can, get this out there to patients who are particularly not really thinking about infection, not really thinking about, because this isn't, they're patients that don't have a medical condition. They're simply using a consumer product, and even though it can be explained to them that there are risks, sometimes people don't always take that message on board, and they can get distracted, and that's when these problems can arise. So if we can have a safety net there that protects that group, then we could potentially have a big impact on those infections. infections.
Your eye is very important, and they don't realise that this is a medical device that they're putting into their eye. It is a fashion thing at the end of the day, and as you were saying as well, there are some people that need to wear a contact lens, but if it's that educating them, this is what you're supposed to do to look after your eyes. It's really important.
Anecdotally, some people even use tap water to rinse contact lenses rather
just
than
to save the money.
Yeah. They assume that's good enough. It's sort of washing them. So but very dangerous. So So again, our sensor potentially could catch that, because if you did that very quickly, you'd get colorization. Our sensor would go off and warn people.
So in the biosensor world, is this one of the first consumer type products that are entering this space? I know there's biosensors out there for all sorts of things, but I haven't been aware of one that has actually made it to consumer type products. So
I think you're right. I mean, certainly we've been looking at what other approaches people have taken to recognizing and detecting infection on medical devices, and typically that tends to be much more expensive, complex kind of approaches. So people might, swab and have rapid kind of PCR methods for detecting and genotyping organisms and so on, but which is great for research and might be suitable in a very kind of medical environment, but for consumer products it's not really an appropriate approach. So we're not aware of any other technologies like this. That there was an approach to develop a contact lens case that would dissolve. So after a month it would just sort of fade away, so that would stop people overusing cases, but I think that never really took off. I guess there are issues around that. So.
All right, if anyone has a question, please feel free to come up to the microphone. Do we have any from the Twitterverse? Looks like they're shaking their heads, so we have no questions. How about the audience? Do we have a question for our two authors here this morning? If not, I'd like to thank my two guests.
I think we have
questions. That is good.
Alexei Gerasudo from PTC Therapeutics. Very interesting idea and cool product. Just a question about how broad is the pathogen coverage for your device, and kind of like maybe you can comment on sensitivity in terms of like, does it actually change from yellow to blue? Is there intermediate color changes or things which you actually detect in the device?
Sure.
All good questions.
Sure. So, I mean, I think we've alluded to the fact that the device doesn't trigger with viral infection. So it is essentially bacterial and fungal. In fact, we've focused the initial testing on the ISO standard, IC 14729, which is the standard for multipurpose solution testing. So it was sort of we zoned in on the organisms that are the same ones that you test when you're testing the efficacy of multipurpose solutions for use with contact lenses. But outside of that, we've now tested 20 other organisms, and we get pretty much identical results across the board. And I think it's because we're using some fairly robust tetrazolium chemistry. It's a metabolic indicator. So, it's something which is not likely to vary between organisms. The only thing it doesn't work with are viruses for obvious reasons. So in terms of, do you want to talk about the threshold?
So you mentioned about the colour change. It really is an all or nothing colour change. So we can set that threshold. In this case, we wanted it to be around 10 to the 4 CFU per mil. We think that's clinically relevant, and above that, anyway, the end user would see this cloudy solution. Below that, it's not really a problem. So we've set it just at that level, and that is when you get this colour change, and there's no in-between colour change.
That's right. That's right.
So the spectrum of what about the precision of your test? Can this case be reused over and over, or will the tetrazolium chemistry steal enough electrons from last night's use until tonight's use, and you'll actually get a false positive?
Sure. So we've designed the technology with that in mind. So it's designed to be reused over the course of a month. Beyond that, we're not certain, although of course you shouldn't be using your case beyond that. So actually it wouldn't really matter if it triggered, because it might be a prompt to say stop using your case. So, but essentially, yes, it's designed for that. I mean, there are other things we've had to look at. So we had to make sure that multipurpose solutions themselves didn't trigger the chemistry. But we confirmed that, because with a range of solutions from peroxide through to other MPSs, the standard MPSs. So yeah, I think there are those issues, but that's certainly something that we've kept in mind. A lot of the research has been around that and making sure that it is suitable for those sort of timescales.
Did we miss anything in our questions of you here this morning? Would you like to add anything else?
No, I think we've covered everything. Thank you very much.
I think so, yeah. I mean, I think the only other thing, just to set this into context, is that the same technology, as I mentioned, is being developed for other areas. So we have some other products in the pipeline, which maybe we'll be coming along to future conferences with, which are in the areas, for example, of dialysis and wound care, where again there are major problems, and in fact, in some ways, bigger problems still. Where certainly
It's life-threatening.
indeed, where people have serious medical conditions that can be, if they have an infection, essentially they're not going to recover from. So
But the good news is, by going to consumer product first, you'll hopefully drive those costs down, so we can actually incorporate them into clinical care at a very inexpensive price point.
Exactly that. Yeah, no, that's right. I think we're very hopeful we can do that.
Yeah.
So, yeah.
So I'd like to thank my guests, Dr. Curtis Dobson and Dr. Nishal Govindji-Bhatt, for joining us here this morning at ASM Live here at the 55th InterScience Conference on Antimicrobial Agents and Chemotherapy here in San Diego. Please join us again at 1 p.m. Pacific Time, where we'll have another guest at ASM Live. And if you care to review this session or any of our other sessions, they're archived for you at ASM's site, microbeworld.org, and you can listen to this again, as well as all of our podcasts, TWIM, TWIV, and TWIP, and the newest one, Microbe Magazine. So until next time, listen for us on the podcast at This Week in Microbiology, and I'm Michael Schmidt, and thank you for your time. Thank you.
Thank you, Michael. Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.